Great.
And so, Michelle, as the leader of BHS, it's my great pleasure, privilege to hand it off to you.
Great. Yeah, thanks, Karen. Yeah, this has been a highlight to us, and in some ways born out of a degree of necessity. I think when we started this, it was a pragmatic solution to a conflict with my course at the same time as the RIP. And actually with Brian and your help, and honestly, I'll do a shout out to Vijay and Ciprian's team. And that's how the genesis of this really was finding another time where we could talk, and there was so much momentum, as you said, around this building area of technology. Vadim, I think, was just an assistant professor at that time. And so it was just a lot of fun to kind of start there and see how much it's grown. And of course, with Jennifer's group, it's really exciting to see. I think in just 7 years, 8 years at the most, how much it's grown. So just wanted to reinforce, sometimes things happen in funny ways that turn out to be gifts to us all. So I thank you for making this time for us on a Tuesday afternoon as well. So with that, I will introduce my team. And I think our agenda is really focused on the wearable technologies that we've been spending our time on, really fairly really building over a series of years. And what I'll do is introduce the members of the team, most of whom will be speaking today, I believe. As I go across the room, we have Breanna and Pat, both of whom are EBA pre-doctoral fellows in the Department of Mental Health with me. We have my team on the staff, Dana Eldrith, Dr. Dana Eldrith and Emily Richards, who are here with us today. And then my, thank you. Yeah. And my practicum student, who's actually been my MPH student as a part-time, he has a full-time job. He's a father, a new father, and he's doing his practicum and his capstone all, I don't know how he's doing it, and bought a new home. And so that's Danny Scerpella. If you want to, you'll meet Danny. Some of you may know him as well already. So that, I believe, comprises the members who will be kind of tag teaming today, because the work that we're presenting it's been a real team effort, and it's really succeeded despite challenges of COVID and the pandemic that have really limited our progress until recently. So with that, I'm going to pass the baton.
Thank you, Michelle. I'll share my screen now. Okay, can everyone see the presentation? Okay, great. I'll let Breanna begin it now.
Sure. And thanks, Dr. Carlson and Dr. Bandine Roche, for that introduction. So today we're going to be presenting our talk titled Validating Smartwatch Technology to ActiGraph, Measuring Accelerometry and GPS in the Real World. Next slide. So wearable technologies really offer an opportunity to collect passive and objective data, both activity data and location data, to reduce recall bias, potentially reduce participant burden, and also increase the measurement precision from the data that we get. Next slide. So today we'll be presenting on the WAVE study, and WAVE stands for Wearables and Accelerometry Validation in the Environment Study. The goals are to look at the quality of location data from the Apple Watch versus the iPhone in collecting location data. The second goal is to validate accelerometry using a commercially available device and comparing that to a research grade device like the ActiGraph. And ultimately, the goal is to have a synchronized data set with both GPS and accelerometry from one device, so that we don't have to combine timestamps or go through that laborious process. So data collection is currently ongoing. We are anticipating a sample size of around 30 participants. Right now, we have recruited and completed data collection for 11 participants. Their mean age is around 30 years old, mostly female and 64% white. Next slide, please. So for the study procedure for WAVES, participants are asked to wear an Apple Watch and an ActiGraph on their non-dominant wrist for 10 days. They're also asked to carry an iPhone on their person for 10 days. And they were also asked to keep a wear time activity diary, reporting at 1 hour intervals what they were doing during that time frame. And so from these data, we can collect accelerometry. We can also collect location data and step count. Next slide. So for the quality control and analysis, the sampling rate for the 3 devices are listed here. So for the iPhone, the sampling rate was set to 30 seconds. This was done after some pilot testing, and I'm happy to go into more detail later. For the Apple Watch, the frequency was set to 31 hertz. The reason why it wasn't at 30 hertz, there was some competing processing going on the Apple Watch, where when it was set to 30 hertz, it led to these uneven sampling rates. So we had to up it to 31 hertz. And then for the ActiGraph, our sampling rate was 30 hertz. So in order to look at the quality of location data from the Apple Watch versus the iPhone, we downsampled the Apple Watch files from this large amount of data of 31 hertz, 31 times per second, down to 30-second average intervals. And so this has implications for data synchronization so that we can look at the iPhone's 30-second intervals with the Apple Watch's downsampled 30-second intervals. So next slide, please. I'll pass it off to Emily.
Great. Thanks, Breanna. So now I'll go through the 4 steps for our quality control process. So the first is a visual check for data completeness. The second is checking files for any unanticipated formatting that may not be correctable in our script. For example, if there's any pipe delimiters, which I've shown that symbol right here. Our third quality control script is our third quality control step is running our script that Breanna created. So this script checks for semicolons and misaligned headers. Then it creates a combined CSV file. So this file is a big file that has all of the participants' data for their full 10 days into one big data set. Then it takes the average of a 30-second interval period and then aggregates 2 rows for each minute of data. The fourth step is placing the iPhone and the trimmed Apple Watch files into ArcGIS for data visualization. Our first goal is to compare the quality of location data from the Apple Watch versus the iPhone. And here are some of our findings. So this slide shows the big picture of an example of 4 participants' data that we've shown here in ArcGIS. The data, as you can see, is geographically diverse, and our activity footprint is variable, which is great. So each color here represents 1 participant. The darker colors represent the Apple Watch, and the lighter colors represent the iPhone data. We see here that there's many similarities in the data, so we see a lot of overlap, but we see some differences as well. So Danny will continue in the following slides by showing the same data, just displayed a little bit differently for ease of presentation.
All right, thanks, Emily. So yeah, what you're seeing here is the participant data for one of our participants in the trial, and this data represents the full 10-day observation period for this person. For some context looking here, it's labeled here as well, but the red dots are the dots that were collected for the Apple Watch, which have been trimmed to every 30 seconds, and the green are the iPhone data that was also collected at 30 seconds to begin with. So what you can see here is fairly uniform agreement between the 2 devices in terms of the collected data. There's some minor variability that you can spot down there in the southeast of the map where it doesn't quite look like 1-to-1. But for the most part, this data for this participant does seem to suggest what we'd consider a sort of quote unquote best case scenario where the devices are showing relatively similar tracking data. You can go to the next slide, please. All right, so this is another individual participant here. In this one, the magenta dots are the Apple Watch data and the green dots are the iPhone. So as you can see here, there's some very evident incongruity between both devices. That's evidenced here by, you can see a lot of the green dots are not overlapped by a magenta dot, which shows that the Apple Watch was not collecting data in these areas. Emily, if you can click through real quick. All right, so on this close-up here, what we did is we changed the map to show some major roadways. And so we highlight with the circles the incongruities, a couple of them that you could see from the larger image. So what you can note here is that the incongruous iPhone tracking data follows major roadways for the most part, suggesting that these data were collected correctly and it's not erroneous data that's firing off like as a mistake. Like these are following roadways and therefore seem like they're legitimate tracking data. One thing I want to point out is the circle towards the top. We want you to take special note of those dots as they are on a very specific roadway that you're going to see repeated in the next image. And we'll talk about the implications for that as well. So if you want to go to the next slide, please. Okay, so this is another participant again, this time just again for context, the green dots represent the Apple Watch data, the red are the iPhone. So you can see here there's a very pronounced like sort of southern trail there at the bottom of iPhone tracking data that's not reflected from the Apple Watch data. You know, this could be for a number of reasons, but we're going to focus actually up towards the top of the image in the next slide. So Emily, if you can go ahead one. Okay, so again, we bring in the major roadways here. So in this close-up, you can see that, again, the iPhone data seems to follow the major roadways and doesn't seem to be erroneously collected. So again, we're seeing iPhone data following roadways where there is no Apple Watch data following. So looking at that northeast corner of the image to the right there with the circle, this represents similar data to the previous image. It's the same sort of major roadway there. It's not quite the same, but it's a little further down. But we did find it interesting to note that we were seeing an Apple Watch dropout for the signal in the same sort of area across two different participants. So it may suggest that there's some linking factor between the two that can explain why the Apple Watch is dropping out. But it does warrant further investigation, as this is really just looking at two of the participants that we've collected. So ultimately, what we'll need to do is break the data up into the individual days of tracking for each participant and just make note of any of the participant offered explanations. So it could be in the self-report study journal that they mentioned that, you know, the devices were charging or they forgot it at home, the Apple Watch. There could be a lot of explanations to why this data is happening, but it is an interesting note there. It is interesting. One other thing to note is that one thing we have noticed across a lot of this data is that when there are incongruities, it's usually the iPhone tracking data that shows up without overlapping Apple Watch data. And that seems to be pretty consistent, that for some reason, the iPhone seems to have data where the watch isn't gathering it. So it's just something to take note of. Next slide, please, Emily. All right, so this last part is another participant with tracking data between the Apple Watch and the iPhone that seemed to be in relative agreement. This is closer to that sort of best case scenario we were talking about. The reason that we're sharing this image is because it represents sort of an early analysis that we've been doing in ArcGIS Pro, which has kind of revealed sort of an interesting pattern that may have some potential future utility. We're still exploring. Specifically, what we did is we analyzed the data for points which were identical between the two devices. That's represented by the triangles on this map. So the instances of the overlap are not as widespread as you'd expect, considering these two devices were being worn together over 10 days. At least I would have expected to see a little bit more overlapping data points. But going into those overlapping data points and looking at the underlying data, we realized what it shows is that these data points only exist in places where participants were in the same location for at least a minute without seeming to make any large movements that would have pinged a different data point in a different spot. So while some of these are only showing like a spot of like sitting still or being in one place for a minute, examining some of the others show that there's no motion for hours at a time. So what we're thinking here is that these sort of these data points or these aggregated points that we're looking at, or these overlapping points, I should say, may potentially suggest that we can quickly identify areas where a patient's in one location and not seeming to move very much. We can't comment on the level of activity of these people, that we know they're in one location, that the devices at least are not moving. But it is an interesting way to see that this might be a place where people are spending a lot of time, regardless of what activity is happening in those. One very important thing to note is that these hotspots could be a result of the participant just not wearing the devices, that they're charging, they could be sleeping. There's a lot of reasons why these things are showing up. And we'll obviously be validating these data points with the ActiGraph data that we have as we further integrate that data. So next slide. There's two more here going here. So some of the things that we've learned so far is that there are potentially geographic dead zones that might relate to some of the data gaps that we see, specifically the missing data on the Apple Watch, on those major roadways. Some of the reasons that we identified why this could be happening: there could be signal dropout, the battery could be dying, we could be seeing instances of urban canyoning, there could be sensor differences between the two devices. These are all things that warrant further investigation as we get more data and we dig a little bit deeper. The other thing that was worth noting that we've learned is that the size of the data files from the Apple Watch are very large, to the point where there's hundreds of thousands of data points per person when you're looking at the data combined over 10 days. So it's very unwieldy for doing any sort of quality checking on the data. Also, it's not necessary for the broad comparisons that we're doing here. So, just showing the overlap of data every 30 seconds are pretty useful for this. And then finally, there might be some potential difficulty interpreting significant findings from the noise, especially as it pertains, as we bring in the activity data that was collected on the Apple Watch. Having so many data points might sort of even out any peaks that we're looking for in terms of activity as we get into that. Next slide. And then just lastly, some challenges that we want to note is that the programs that we're getting the data off of the iPhone and the Apple Watch seem to have some inconsistency in the data formatting that doesn't quite follow what we would expect, in the ways that it just comes out inconsistently from time to time. Sometimes it has comma delimiters, sometimes it's semicolon. We're able to address these things fairly quickly, but it's just something we have to watch out for. A huge challenge with this kind of work is data privacy. We have to be making sure that we're careful stewards of this data. It's very identifying for people. People are giving us access to a lot of their locations. So as we demonstrate a lot of this data, we try to make sure we're pulled far enough back that it would be very difficult to identify specifics of what's going on with these people. And then finally, one of our challenges is just we're thinking of ways to combine our data, our GPS data elements with some of the metrics, such as our actigraphy and across time. So like those are some of the things that we have to parse out to maybe get at some more intricate lessons learned. I think that's just my part.
All right, so I'm going to highlight some of our steps that we've been working through for the validation of the Apple Watch and monitoring movement in the real world. Next slide, please. All right, so our goal here is we're trying to validate the accelerometry from a commercially available device, which in this case is the Apple Watch, to a research grade device, which in this case we're using the ActiGraph GT9X. Both are being worn on the participant's wrist. And we want to evaluate the accelerometry and the GPS data that's being collected from one device. So in that case, this is the Apple Watch, whereas an ActiGraph does not collect GPS data. So we're hoping to be able to get both of these types of data from using only one device. Next slide. So this is kind of our outline of some of the preliminary steps that we've been looking at so far for processing the Apple Watch data. So we've taken the three axes of the accelerometer, the X, Y, and Z axes, and calculated vector magnitude units using the raw data here. And then calculate the average vector magnitudes for each 60-second interval on the Apple Watch. And then our next goal, it would be to examine the correlation between the Apple Watch and the ActiGraph vector magnitude units. And we can look at this at the minute level, hour level, day level, etc. Next slide. So this is a very rough overview. We looked at 2 different participants and 1 minute's worth of data for each participant. And just for reference, 1 minute's worth of data is about 1,800 data points here, because we're sampling at 30 Hertz or 31 Hertz, depending on the device. So this is showing here participant 1, when they wore the ActiGraph and the Apple Watch for the same consecutive minute, what their average vector magnitude in Gs was measured. You can see pretty similar values here with participant 1 and with participant 2. Participant 1 was a little bit higher, so potentially some more movement going on with them, which also may explain why their standard deviation is a little bit higher as well. but overall pretty similar values, which is a good sign. Next slide, please. So some of the challenges that we've been experiencing with this data is whether we want to continue to use Gs from the Apple Watch, which is how it's collected using the app that we're using at least, or whether we should convert these values into what ActiGraph uses, which is known as counts. And part of the reason for this is there's a low variation in the Gs that we've been seeing, at least so far, in the participants that we've looked at. So it kind of makes it hard to distinguish whether people, for interpretation purposes, like how active are these people when there's such very small differences in the actual numbers. There's also been gaps in data for the Apple Watch, and this is most likely due to battery issues. The Apple Watch battery doesn't last that long, where the ActiGraph battery can last for a couple weeks. So lining up these timestamps to make sure that we're analyzing the exact same minutes of time that the participant was wearing the device can be a little bit challenging. Next slide, please. So some of our next steps that we're thinking about is we want to be able to link the individual activity levels to the environment overall. So we want to determine the feasibility of using this commercial device for both GPS and accelerometry. And we want to be able to link the individual accelerometer data with group level environment data, and that's using ArcGIS that we've gone over in some of the earlier slides. And to answer some of these questions, like, is time spent outside the home related to your health? And are there different physical activity hotspots in the community? And we'd also like to explore census and block level indices of neighborhood level characteristics like SES that may account for activity patterns and health. Next slide, please. And these are some examples of what we're hoping to do. This is actually from previous work, but on the left here is a map showing some of the activity hotspots. So the kind of yellow color to red is showing the clustering. So that's like if someone was spending a lot of time in a certain location, that's going to have that darker red color. And then the actual vertical bars there is the step count. So higher bars would indicate greater steps that are being taken in these areas. And then on the right side, that's just a figure showing the amount of time spent geographically in different regions, with darker colors indicating more time spent in those regions. Next slide, please. And so I'd like to thank Dr. Carlson and the rest of our lab here and the Epidemiology and Biostatistics of Aging program and our training grant that supports me and Breanna. And that is all we have. So any questions?
That's fabulous, first of all, you guys. And so we are low on time for questions. I'm going to quick throw out a couple from the chat that were sort of repeated. And so the one had to do with whether the Apple Watch is sort of piggybacking on the iPhone, you know, and then that might be why the iPhone is the dominant of the two, you know, so partially using the iPhone to draw data.
I can answer that. So yes, when the phone is paired with the watch, some of the offloading and the processing goes through the phone. But the watch itself does have a GPS. So if you leave your phone at home or leave it in the car, your watch will still be able to collect those data singularly without the phone necessary nearby.
Okay, and then another one that I saw sort of repeated has to do with equity of positioning and such, you know, so were there cases where one of the devices, say the watch was being worn and the iPhone was sitting in a, you know, a charger, you know, and with the ActiGraph and the Apple Watch, were they being worn on the same wrist? Was one sort of higher on the arm than the other? Those sorts of things.
Emily, did you want to answer about how they were worn?
Sure, yeah, I can do that. So both the Apple Watch and the ActiGraph were worn on the non-dominant wrist, and they were right next to each other. So the positioning was basically as similar as we could between those two.
And just to follow up on that, if they are next to each other, they are actually subject to different centripetal forces, because one is closer to the elbow than the other. So you will have correlation, but you will not have the same thing, just because, you know, if you are closer to the wrist, you will have higher forces than if you are farther away. So this is the type of thing. So one thing that you may want to check. So this goes back to a discussion we had a long time ago about how to compare these things. It's very difficult. One of the things that one could do is to actually try not in humans, but in robots, right? So we take a robot and we do the same movement. And then the other closest possibility would be to have them taped on top of each other, even though that still doesn't solve the problem, because then you have an additional component in one direction versus the other. So correlation, but not identity.
Yeah, those are great points, Chipper. And I remember the early days it was movelets, but I remember you guys presented and showed how it showed us real-time data. No, it's great points. I think for us, we're going to see if we can get close, recognizing that we're probably not measuring exactly the same thing, but looking at as from Pat's data, you saw the correlations. We were actually kind of surprised at how high the correlations are, but we'll have to keep in mind, unfortunately, taping the two together in our everyday lifestyle, participants probably won't be feasible, but it would be a specific kind of test we could do amongst ourselves, you know, kind of do a little bit of research work amongst ourselves to try to put them on top of one another and see what the data look like. So yeah, it's a great idea. And I did want to return to something, perhaps we can keep it up. There's been some great chat questions. And I think that what's also important to note is that our goal will be to really see if we can use these commercially available devices that allow us to synchronize both GPS and actigraphy. That's our overarching goal, is to kind of simplify. And it, you know, I think right now the jury is out on whether or not the Apple Watch will do that for us. There are just all the things that have been mentioned and things you're bringing up in the discussion make this a continuing kind of an effort. So there's more to be done. So yeah, thank you all.
Thanks for a great discussion, everyone. So if the BHS folks can stop sharing your screen, and we can pass on to Engage now. And for this, I'm going to pass on to the eminent Engage leader, Jennifer Schrack.
Wow, I don't know if I deserve such a big title, but thanks, Karen. Thanks, everybody. I'm just going to really quick introduce our speakers, so not to take away time from them. So we have Francesca Marino, a second-year PhD student EPI, who's very interested in physical activity and brain health. She works closely with our group, and her advisors are Amal Winigatunga and Jennifer Deal. And then Fangyu Liu, who is also a second-year PhD student, who's been with us for a while as a master's student, a research assistant, and now as a PhD student, who's working on some data from the SPRING study, the study of physical resilience in geriatrics, of whom several of the people on this call are investigators. So I'm sure they'll be excited to look at some of that data. So I'm going to turn it over to Francesca to get started.
Okay. Are you all seeing the right orientation of the slides? Yes, okay. Great. Thank you, Jen, for the introduction. So today I will be discussing my work, looking at the associations between Alzheimer's disease biomarkers and daily physical activity patterns. So dementia affects nearly 50 million individuals worldwide, and the prevalence is expected to increase to 152 million over the next 30 years as the population continues to age. Alzheimer's disease is thought to be the most common form of dementia and is the focus of this work. Alzheimer's progresses along a continuum where there is a long preclinical phase that can last for decades. It is important to detect individuals during this stage because it is thought that this is when interventions to prevent or slow the decline may be most effective. This figure shows different biomarkers for beta amyloid and tau, which are the hallmark features of Alzheimer's. As you can see by the colored lines, the concentrations start increasing during the preclinical phase, many years before an individual develops cognitive symptoms. And today I will focus on beta amyloid, which is indicated by the red line on this figure, and also the ApoE4 genotype, which confers increased risk of Alzheimer's disease through mechanisms relating to beta amyloid. So given the importance of early detection, it is important to determine whether there may be other markers that are associated with these existing biomarkers of early impairment. There is established evidence for the benefits of physical activity and exercise on cognitive function. Potential mechanisms for this may include increased neurogenesis, cerebral blood flow, and brain neurotrophic factors. A limitation of the existing work in this area is that most research has focused on exercise and has also relied on self-reported measurements. So today I will focus on daily physical activity as measured by wrist accelerometry, as we've just heard a lot about. So as we know, accelerometers provide a method to monitor an individual's daily movement throughout their routine environment. And this allows us to measure daily physical activity, which is any movement resulting in energy expenditure and not just planned exercises. And accelerometry data can be processed into specific patterns, which give us information about how someone is moving throughout their daily environment. Prior work has shown that some daily physical activity patterns were more strongly associated with cognitive function than summary measures of total activity. And wrist accelerometry also measures 24-hour rhythms and both upper and lower body movements, which may better capture Alzheimer's-like behaviors such as sleep disruption, agitation, and wandering. We know from previous work that increased physical activity is associated with lower production and increased clearance of beta amyloid, and also better brain structure. And presence of Alzheimer's biomarkers is also associated with slower gait speed. So given this background, our overarching hypothesis is that daily physical activity patterns can capture differences in daily movement as a potential warning sign of Alzheimer's pathology. For these analysis, I used data from the Baltimore Longitudinal Study of Aging, which is an ongoing study of human aging that began in the 1950s. Participants were initially free of cognitive and physical impairment on enrollment, and the BLSA has an age-dependent follow-up schedule where older adults are seen more frequently to better capture age-related changes. And in this analysis, I restricted to participants aged 55 years or greater with data on Alzheimer's biomarkers and wrist accelerometry from the same visit. In 2015, the BLSA initiated wrist accelerometry data collection using the ActiGraph. And these data, again, are processed into daily physical activity patterns. Today, I will focus on total activity counts, which reflect the amount of activity. And the Alzheimer's biomarkers of interest in these analyses include elevated beta amyloid, as measured by Pittsburgh compound B positivity on PET imaging, and the ApoE4 carrier status. And we fitted linear regression models to regress total activity counts separately on each biomarker. So for sample characteristics, overall, we see that individuals in each exposure category are pretty similar. We do see that PIB positive individuals, where again, PIB is referring to elevated beta amyloid, but these individuals are generally older, have lower self-reported health, lower physical function, slower usual gait speed, and report more disability and activities of daily living, although many of these findings are non-significant due to small sample size. And we also see that ApoE4 carriers are generally younger and are more likely to be of Black race. So I first examined differences in total activity counts by PIB status. And this figure shows the diurnal curve of average total activity counts over 24 hours, where higher peaks indicate higher activity counts. So we see a peak in activity in the morning and afternoon, and a sharp decline in activity after around 6 p.m. And PIB positive individuals in green tend to have lower activity during daytime hours, but these curves are very close together and are largely overlapping, which suggests non-significant differences in total activity counts by PIB status. So I then fitted linear regression models to examine differences in discrete 6-hour time bins of total activity counts by PIB status. The models were sequentially adjusted for additional covariates, where model 3 is adjusted for age, sex, race, education, and gait speed. And overall, we did not find any significant differences in total activity counts by PIB status for any of these time of day categories. And to interpret a coefficient, we would say that for PIB positive individuals had on average about 2,800 higher activity counts between midnight and 6 a.m. as compared to PIB negative individuals after adjusting for the covariates in this model. So we then examined potential effect modification by age by including an interaction term between PIB status and continuous age to the same models presented on the previous slide. So the first column shows the unadjusted model without an interaction term, and the next three columns show the interaction regression coefficient. So here we see that for each 1 year in age, the average total activity counts between midnight and 6 a.m. were about 3,400 counts higher among PIB positive as compared to PIB negative individuals. And this shows that with increasing age, PIB positive individuals tended to be more active overnight and in early morning and evening hours, and trended towards having lower activity during mid-morning hours as compared to PIB negative individuals. So we then conducted a similar analysis looking at ApoE as the exposure. Where here we are again looking at the diurnal curve of average total activity counts. And again, these curves are largely overlapping, which suggests non-significant differences by E4 carrier status. And this was again confirmed when looking at linear regression models where we did not see any differences in total activity counts over any of the time bins by E4 status. And then again, we examined effect modification by age. And we see here that for each 1 year in age, E4 carriers tended to have about 1,555 fewer activity counts between midnight and 6 a.m. as compared to non-carriers. And this shows that with increasing age, E4 carriers tended to have lower activity overnight and in early morning as compared to E4 non-carriers. So in conclusion, we found that with increasing age, PIB positive individuals tended to have more activity overnight and in early morning and evening hours, and trended towards less activity during mid-morning. This suggests that older PIB positive individuals may be more active overnight, which could be reflective of sleep disruption or Alzheimer's-like behaviors, such as agitation or restlessness. And we also found that again, with increasing age, E4 carriers had less activity overnight and in early morning. And when we looked at specific age margins, we found that this was non-significant among the oldest old at ages 80 and 90. And this may reflect possible decreases in the predictive power of ApoE4 with increasing age, which is a finding that we know from the literature. And again, these findings are cross-sectional in nature, so further work is needed, but interesting initial findings. And I'm happy to take any questions, or can also stop and let Songyu go if you would prefer.
Do you want to take one or two quick questions? Andrew was asking about the models. I think I confused him. So did you get your answer, Andrew?
I actually, I'm not sure that I did, but Vadim, I think, had a great suggestion to do either a function on scalar or scalar on function regression model.
If you can give me a good interpretation of the outcome, we'd be happy to try that.
Yeah.
The problem with function, and I'll leave that up to the experts in the room, but the problem I've always had with functional regression is the interpretation. So
it's the same with what you're doing, is just more beams.
But it can't actually quantify the exact difference with what I was told before.
Same exact thing, is just more beams. So I think this is great. We can talk about that, but there is no difference between the two approaches. Practical difference.
Okay. And I will say, just to note, we did look at 1 hour bins as well. The tables get very large, so I didn't present them here. It's overall the same conclusions, but yeah, we've talked about that method before, and I'm happy to learn more about it and explore that as well. Okay.
This is wonderful. I think that I'll try to do this to reproduce your results in UK Biobank and see if we can. You can reproduce them, because we have a lot of people there, and it would be interesting to see.
Yeah, that would be great. Sample size is definitely an issue, especially with the PIB sample here. It's quite small.
I would also add that in some of the previous work, we investigated looking at the curves which represent not just average activity at this specific time, but actually variability at the subject level. For example, if you have 7 days, on the same subject, you look at a certain time period and you calculate how variable subject activity during this period, and you connect this as a curve. So essentially what you have a variability curve, right? And then those actually show much more significant. I'm trying to find the paper we published, I just couldn't find it somehow. But I'll send you the link in the chat, so you get the sense like what And would be very happy to share the code, so you could just like run it immediately. Just see.
Yeah, that would be great. Thank you so much. This is part of my dissertation work, so I'm really interested in looking at complexity and variability of activity as well, and kind of moving just beyond amount of activity. Thank you, though. I appreciate that a lot. Okay. I can stop, so you can share.
Okay, Fangyu, you want to take over?
So hi, everyone. Today I'm going to present some descriptive data we get from the SPRING study. Can't move. Okay, the SPRING study is designed to study physical resiliency in older adults in response to a stressor. It has 3 stressor projects. The first one is total knee replacement surgery. We call it a RESTORE study. The second one is bone marrow transplant, and then from now on we'll refer to as rebound. And the last one is initiation of hemodialysis or READY study. So the motivation of looking at the novel metrics is that physical activity was originally planned to be a phenotypic measure to measure whether a person is resilient or not. So we want to see how the physical activity metrics changes before and after the stressor. However, due to the time series nature of the accelerometer data, we were wondering if they can be served as a dynamic measure, similar to oral glucose tolerance test or heart rate variability. If they can serve as a dynamic measure, maybe they can be used to predict one's resiliency before the stressor happens. And then the second reason is that we want to understand physical activity that's beyond the total activity. Therefore, for these 2 reasons, we explored some novel metrics summarizing the accelerometer data in the SPRING study as a first step. So we explored 2 sets of novel metrics. The first set we refer to here as the physical activity pattern metrics. There are 4 groups of variables. The first group contains the mean activity counts in the most active consecutive minutes of various window lengths. The second group looks at the mean activity counts in the least active consecutive minutes of the same length. The third and fourth groups differ from the first 2 groups in that they do not require the minutes to be consecutive. So the activity count is almost 0 for everyone, even when using the 480 minutes window for the fourth group. Therefore, we will not discuss the fourth group today. The second set of metrics we look at are circadian rhythm variables that include first acro time, that is a measure of the timing of the maximum activity, assuming a consignorship of the data. The second one is pseudo F statistic. It is a statistic of the fit of activity data to the cosine function, indicating the rhythm robustness. The third variable we look at is intra variability. It is a measure of the fragmentation of the rhythm relative to its 24-hour amplitude. So higher value is usually interpreted as daytime napping and or nighttime arousal. The last one is interdaily stability, which measures the stability of the activity profiles over days. A higher value of the interday stability means a higher day-to-day variation. Next, I will show some descriptive plots of all these metrics. On this slide, I'm showing the most active consecutive minutes. I picked the shortest and the longest window and the 2 other windows in between. The red lines represent the RESTORE study, that is the knee replacement surgery, and the blue lines represent rebound, which is the bone marrow transplant. The RESTORE study have both data points at 4 months and 6 months because we are using data from phase 1 and phase 2 of the study. And in phase 1, participants are scheduled back to come back at 4 months, whereas in phase 2, people are coming back, follow up at 6 months. So when put them together, we have 2 data points at 2 different two different times. Regardless of the window length, we can see there's a slightly, so for restore, we can see a slightly decline after the knee surgery at 1 month, regardless of the window length. But the decline is much more prominent after bone marrow transplant in rebound. We also see a recovery of the activity level at 6 months in rebound, almost back to the level at the baseline. There are a few participants with accelerometer data for the READY study. So on the graph, we only have one green dot that represents the baseline level of the participants for these measurements. Next, this slide shows the plots for the least active consecutive minutes. Mean activity counts in consecutive minutes less than 60 minutes is essentially 0 for everyone. So I'm showing here the plots for 60 minutes, 90 minutes, 240 minutes, and 480 minutes. It may seem there is an M shape for the restore study, but remember participants who had 4 months data and those who had 6 months data are completely different. The plots, except for the 480 minutes on the bottom right, suggest that in both restore and rebound, mean activity counts in the least active minutes increased at 1 month after the surgery. Since the least active minutes are most likely during the sleep time, this may suggest sleep disturbance right after the surgery. The other thing to notice is that recovery to baseline level was not as prominent as in the previous slide. So at 6 months for both studies, we don't see the measurement recovers to the baseline level. This slide shows the most active minutes. So here the minutes do not require to be consecutive. It's just the top like most active minutes throughout the day and aggregate together. We see a very similar shape of the plots as in the case where we have consecutive minutes. The last slide here, I have the 4 circadian rhythm plots. On the top left, we have the accrual time. That is the timing of the maximum activity. Rebound and restore have the opposite trend for some reason. I find this very interesting. For rebound, the timing of the maximum activity becomes slightly earlier in the day at 1 month, and then becomes later in the day at 6 months. But for restore, the timing of the maximum activity becomes slightly later at 1 month, but becomes earlier at 6 months. If we move on to the plots here, so for the pseudo F statistics, we see a decrease in the fit to the cosine function, meaning a less robust circadian rhythm at 1 month, and a recovery at 6 months for both rebound and restore. But the decrease is less prominent for restore, and the recovery is well beyond the baseline for rebound. For the intra-daily variability, we see participants in rebound had much worse intra-daily variability level at 1 month compared to restore participants, but they recover at 6 months. Though higher intraday variability is usually interpreted as daytime napping or nighttime arousal, in this patient population, it may actually mean like weakness after the transplant surgery or bad rest for recovery after the surgery. Lastly, for the interday stability, lower value means higher day-to-day variations. Participants in both studies deteriorate after surgery in terms of the variation, but recovers at 6 months. So the overall take-home message is that we see, in general, there is a deterioration for all these metrics at the 1 month after surgery in both studies. The deterioration is in general much stronger in rebound. That may indicate in my mind a stronger, like a greater stressor comparing a bone marrow transplant to a knee replacement surgery. And third point I want to highlight is that recovery or even some improvement has been observed at 6 months for both studies. So the next step for the analysis is that we may want to see some associations between these novel physical activity metrics with other phenotype measures or even clinical measures. For example, like SPPB score, like bodily pain, and maybe clinical outcomes that's relevant for categorizing resilience. That is yet to be done so far, but it's in the plan. That's all I have. I'm happy to take any questions.
That's wonderful, Donahue. I think we have time for one quick question if somebody would like to ask.
I just want to say it's fantastic work. It's fantastic work to see that, you know, what we've been saying, that objective physical activity does the right thing. And you see this in one after the other, right? So if somebody has surgery, their objective physical activity goes down and then rebounds. It's just fantastic to see that. And yeah, thanks.
Yeah, I find the results are very interesting. I'm just reading the chat. I see there is a suggestion from Vadim that I should use like violin plots or box plots. That's a very good suggestion. I will see how the distribution changes with those plots.
Yeah, and you can also switch to more robust statistics like medians and stuff, something like that, you know, just to, because indeed some of these measurements can be very, very skewed, and we have seen that with, and they sometimes can lead to unexpected results. In your case, they don't, but sometimes when you have smaller signals, they could.
Yeah, I just want to quickly add, I think I completely agree with everyone. It's a very cool work, but just be a little bit, I will be cautious about using the Cassiner model. So Cassiner model has been suggested in the literature, but we do know very well it's not working very well. Like, it's not really clear what it's capturing. It's capturing something, right? And also, I even published on this work. Yeah, but it's extremely sensitive to other things, you know, like whether you like log transform your data or not, etc. So I would just quickly suggest that if you're already using like maximal measures, and if you want to capture some sort of a temporality or timing, like a preference for timing for people, I would just suggest to use midpoint of M10 and L5, which is would be much more consistent with the use of maximal measures, right? And that would be kind of multi-resolution analysis. Because Cassiner, again, definitely you could use it, but it's just I don't know, like it's just like more like inertia in literature. People keep using this model suggested in the 90s, which again, much more appropriate for modeling physiological rhythms, like you know, core body temperature or something like that.
Yeah. Okay. Noted. Yeah, that suggestion of the midpoint is very helpful.
Well, why don't we go ahead and thank both Francesca and Donahue for a couple of fabulous presentations, and now go on to the SMART, sorry about that, SMART/WIT group presentations, which has been fantastically led over these years by Ciprian and Vadim, and you know, Andrew, of course, is an alumnus who you know, now is evolving into a leader as well. So we're so proud of everything you're doing. I guess I'll turn it over to Ciprian.
I'll turn it over to Vadim, just because Rahul is his student, and then he's also been leading the group. I'm co-leading it.
And I also turn it to Andrew. Yeah, let me introduce, thank you, Ciprian. Let me introduce two speakers on behalf of our group. So the first speaker is, I think, Rahul Gosal, who will be talking about recent work related also to Alzheimer's disease and kind of looking at the distributional properties. And the second speaker is Erija, who will be talking about a very interesting, I think, functional models for the use in modeling actigraphy. And maybe Ciprian can tell you a little bit more, because I was not really involved in that work.
Yeah, I think that's it's fair to say that a lot of our work has been more recently focused on very large available data sets, NHANES and UK Biobank, and I would like to bring this to all of your attention. I know that there is always interest in new studies, but UK Biobank has 200,000 people with accelerometry data. Just in case you have questions related to that or things like that, do not forget if you have that this resource exists, and we are very good at working with it.
So I guess Rahul, it's your turn.
Yeah, sure. Yeah, I will start sharing the screen. Okay. Hi everyone. So first of all, thanks for inviting me to this meeting. It's the first time I'm presenting in this meeting. So today I'll be talking about one of our recent work where we kind of develop a method we call a scalar on time by distribution regression. And we show that how this method can be very useful for modeling wearable data, in particular capturing both the temporal patterns and the distributional patterns, which some of our previous speakers have been pointing out in their work. And we kind of try to capture both these aspects jointly in this work. And we show the application of this work in modeling association between daily living physical activity and cognitive functions in patients with Alzheimer's disease. So our motivation of the work again comes from wearable data. Wearable devices, as we all know, are a very rich source of data, and they provide user-specific continuous data streams, which can then be used for better understanding of the link between human health and human behavior. So as our past speakers have mentioned, that this physical wearable data is often summarized, can be using various scalar summaries, such as, for example, total log activity count, which kind of represents the total volume of activity, various fragmentation indices, such as say minutes of moderate to vigorous intensity physical activity, or some compositional metrics such as active to sedentary transition probability. But often when we talk about these summaries of such continuous stream of data, we sometimes can lose out the temporal or the distributional information that might be present in such rich stream of data. And functional data analysis, as Andrew suggested, is one way to accommodate or account for these temporal variations in the data. And there has been previous works by our group also on how to capture these temporal patterns. And similarly, the distributional pattern, for example, Vadim mentioned the variability curve, or someone is interested in looking at the maximal levels of say physical activity, then another approach to accommodate those ideas is distributional data analysis approach, where you can model those particular measures of interest and try to use those as your covariates or outcomes in your study. And the motivation and the contribution of this work is kind of, can we do these both two approaches jointly? So can we both jointly capture this temporal or the distributional pattern, which might be complementary in some sense, because they can probably capture some unique features of both these two aspects. So as Francesca mentioned in her talk, we are also looking at Alzheimer's disease patients, because many previous literature has shown that early falls and a decline in physical activity can be indicative of, say, mild cognitive impairment. And in this particular study, we had 92 community old participants who were recruited by the University of Kansas Alzheimer's Disease Center. And among them, we had 39 mild AD and 53 controlled participants. And on each of them, we had a GT3 accelerometer placed on their hip, and the data were collected for 24 hours a day for consecutive 7 days. So at the end of this data processing pipeline, we had actually minute level activity counts on each of these participants for a 7-day period. And we also had actually various cognitive scores on them. And these cognitive scores kind of represent how well you are doing in levels of cognition. And these were obtained from the domain of verbal memory, attention, and executive functioning. Now, coming to our sample, it was a relatively older population with mean age around 74 years. And we didn't have any significant difference between the AD and the control group in terms of age, BMI, and VO2 max. But the AD group by design had a smaller number of females compared to the control, and also had lower years of education. So we had to probably control for these factors when we perform our statistical analysis. So coming back to the existing approaches that I mentioned, so one approach, as I mentioned, is looking at some summary level metrics such as stack or some average activity count. And so for example, for this sample, we can calculate that for each of the subject. And if we look at the population level distribution of, say, this average activity count, we get the distribution which is shown here by these violin plots. And it seems like the this control group has slightly higher median physical activity count compared to the AD, but this might not be, again, very statistically significant. Now, when we go to the diurnal pattern of the daily average physical activity count, which is again a profile we saw earlier in Francisca's talk, we get a similar looking pattern where we see that there is a decline in physical activity, particularly in the early morning between 8 to 10 a.m. and again in the late evening in that 4 to 6 p.m. period for the AD group. And similarly, we can look at the sorted activity counts. And here we are looking at essentially the quantile functions of activity. So we are plotting the average quantile function of activity of the control and AD group. And here the difference occurs in kind of relatively higher quantiles around P to 0.68 to 0.9, where it shows kind of the AD group has lower kind of maximum physical activity compared to the controls. So in this work, what we are trying to do, can we capture both this picture 2 and 3 together and kind of jointly capture this phenomena, which might give us some more insights. And just to briefly show you that this each of these physical activity summaries, for example, the average counts, the temporal profile or the distributional profile, can be accommodated in a regression framework using popular methods such as generalized linear model or scalar unfunctional regression framework, where essentially this XIT or QIP can be used as a functional predictor to model any scalar response of interest. But in this work, what we are trying to promote is a data object we called a time by distribution data object for obvious reasons. And we define this data object as a bivariate function of both the time of the day T and the quantile level P. So what we basically do at each time point, we look at all the activity counts surrounding that time point in your data. So this is a window size which you typically take for, say, aggregation for, say, average activity. So for our study, we kind of prefix this as H equal to 10 minutes, but this could be optimized based on what we are trying to model. And then we can calculate this quantity QITP for each T and P in your data for a grid of values of T between 0 to 24 and various quantile levels. So essentially, instead of calculating the diurnal physical activity mean or the SD or the variability curve, we are calculating all the quantile curves now at each time point T. So essentially, that is what it is doing. And now once you do that, so now this becomes a bivariate functional summary of your data, which captures both this temporal and the distributional aspects jointly. And the nice thing is you can get back your summary level metric, for example, the average physical activity count or the total activity count by simply integrating this object over these 2 dimensions. So the most interesting thing that we found is when we plotted these the bivariate surfaces QITP for our control and AD group. So this plot is showing this average surface of QTP, which is a function of these quantile levels and time for the control and AD group. But we should really concentrate on the difference between these 2 groups. And this clearly shows that the largest difference between the control and AD group occurs at this morning period, but it not occurs at their median physical activity level or their moderate physical activity level. It occurs at their kind of maximal physical activity level. So this really shows that it's the maximal physical activity levels that is different between these 2 groups at these particular time points of the day. So we can use this information for modeling purposes and also for targeting time and your level specific interventions. And now this QITP data object, for example, you have a scalar response of interest. For example, this could be cognitive status or the cognitive scores in our case can be modeled using a bivariate scalar regression framework. So this is a well-established method in functional data analysis. I'm not going over details over the estimation. So I will show how this method performs compared to this model 1, 2, and 3, where we are using this average activity count, the temporal profile of diurnal physical activity, and the distributional profile as a competitors. And model 4 is this proposed method using this bivariate time by distribution surface. And what we see when we model cognitive status as a binary random variable using logistic mean function, all of these effects actually are estimated to be significant. But moreover, the phosphorylated AUC clearly shows that this model 4 goes beyond this model 2 and 3 and actually is able to capture some unique features going beyond the temporal and the distribution information. So we see a slight gain here in terms of the average cost validation AUC from the model performance using this time by distribution data object. And in terms of the estimated surface beta TP, which is a two-dimensional surface now, we see this effect is negative largely during these particular time periods of the day and especially at the higher quantile levels, which is what we expected looking at the surface. It really shows that a higher maximal levels of physical activity during this period is associated actually with lower odds of Alzheimer's disease. And next, we actually modeled cognitive scores, which was one of the continuous measurements we had, cognitive scores of attention. And here the improvement was again much more higher. And we look at the cost-validated R-square here to see any definite gain in model performance. And compared to the distributional summary level or the temporal profile, we do see a large gain in cost-validated R-square compared to the either using the distributional profile or using the temporal profile. So again, it shows that it is able to capture some unique aspects of this such wearable data. And again, coming to looking at the estimated effect, we see that it's actually captured a contrast between high levels of physical activity in the morning and the late evening, and just low levels of physical activity surrounding that period. So that is also interesting from a physiological viewpoint, that it's kind of capturing the contrast or the reserve of a person during these time points as the important signal. And so coming to summarize our contribution, so we are here developed a time by distribution data object, which can capture both temporal and local distributional information in wearable data and offer slightly more general interpretation. And there are various other also possible applications we can do with these bivariate surfaces. For example, we have developed time varying L moments, which can be used for clustering, registration purposes, etc. And some of these remain areas of future research based on our current work. And these are the references I have used. And again, thank you again for giving me this opportunity, and I'll be happy to take any questions you might have. Thank you.
Thank you, Rahul. I think there are a couple of questions in the chat. So there is a question from Karen asking essentially, if we see the major difference in the upper quantiles, could it be driven by outliers, which essentially would be there?
Yeah, that is a very good question. So essentially, in the upper quantiles, what we have seen is can be a very large uncertainty associated with estimating those paths. So it's possible that there might be some overfitting that is occurring when capturing those, say, the higher quantile effects. But we really tried cross-validating therefore as our performance metric to see we are not actually overfitting. We see even with cross-validation, we see that those effects are important, and we are like our performance improves beyond the distribution or the temporal level model. So that's how I think we are confident. But yeah, so there is, I think, large uncertainty that associated with estimating the upper quantiles.
Yeah. Thank you.
And I think there are two quick questions. I think from Jennifer, if this is HIP actigraphy, how reliable are overnight estimates? So that's definitely a very good question, and we didn't look into that. So that's probably affecting, when whether people put back the devices in the morning. There is now a question from Amal. So we don't have ApoE4 information in this data set. And what we did, we only, it was a small study, so we only counting and controlling in some sense for age, BMI, and gender, I think.
And years of education, yeah.
Right, I think for the sake of the time, let's move to the next presentation. Thank you, Rahul.
Can you see my screen? Great. So hi, everyone. My name is Jia Cui, and today I'm very happy to present the work as a parametric risk score for physical activity. So first, I want to briefly introduce the data set. As Chipra already mentioned, here we use the National Health and Nutrition Examination Survey, which is in short, we call it NHANES, which is a large prospective ongoing study conducted by the US CDC to assess health and nutritional status of the US population. And here on the left, I'm listing several features of the enhance, which I think is good for research. It is very large and it is still a prospective study. Most of the enhanced data are publicly available and you can find these data on their website. Also, this is a very rich data set where we have both interview data as well as examination data. And also our group created our package led by Andrew, which is called our enhanced data, which would allow you to download the data from their website and organize and process them into an analyzable format. So here in this project, we focus on the physical activity data collected by accelerometers. And in 2003-2006 waves, participants were asked to wear a hip phone device for 7 consecutive days. And after the processing, the data was released in minute levels. For this project, we actually use the summary measures, such as moderate to vigorous physical activity, or in short, MAPA, and ASTP, meaning active to center transition probability. So you can find a list of those summary measures in this paper. So now let me introduce a bit of the scoring systems. So scoring system is actually a very popular concept in many fields, such as the nutrition field. However, it is still a relatively new concept for physical activity. Like what physical activity should I do every day to get a good score, which would indicate good health status. So following this line, there has been very few published work, but one of them was one of the physical activity score was established using their AARP questionnaire data. And so using this questionnaire data, they established the physical activity score of 8 components. So here on the right, I'm showing the meaning of different components. And here is actually the curve of each score component. For example, for moderate to exercise, their established scoring system suggests that we need to have at least 20 metabolic unit hour per week in order to have a relatively good score. However, so this is the advantage of having this scoring system is there's only one scoring system. And once it is developed, it will remain the same across different outcomes and populations. However, one major limitation of the previous work is the established physical activity score. Since they use the questionnaire data, they may suffer from self-reported bias. So the goal of our project is to develop a single semi-parametric scoring system for physical activity. And here, instead of the questionnaire data, we use the accelerometry data from enhanced study, which is a very large study. However, there remains some challenges. For example, the association between the summary measures and physical activity and the health outcome can be nonlinear. And also in this study, we have different populations like the men and the women. And we also have different diseases outcome. So our solution was to propose a generalized partially additive model to build up this scoring system, which extends the generalized partially linear single index model that was published in this famous paper. And the major novelty of our method is to model each physical activity score component as a nonlinear smooth function. So what do I mean by a nonlinear smooth function? So before we introduce the model, first let me introduce some notations. So for simplicity, let's consider one common disease, for example, all-cause mortality among key populations. Very typical choice of different populations like men and women, and let's use K to denote their the population. And for simplicity here, let's use YIK to denote a binary disease outcome. However, our model can be easily applied to outcome of an exponential families. So the framework will still hold. And now let's use XIK to denote the score components, that the components you want to include in your scoring system, and ZIK to denote those non-score components. That are the components that you want to adjust when you build up this scoring system. So here is our proposed semi-parametric risk score. So we use function S to denote the entire score, which is a function of all the score components. And here we actually write it down as a summation of the separate score component functions. So here, Sj is an unspecified smooth function for the J's score component. Okay, and then we build up our overall score. So notice that here, our score does not depend on the population. So this is actually one feature, one advantage of those scoring system is once it is developed, it can be applied to different populations for different diseases. Okay, so this S is not a function dependent on K. So if we look at this model, this is how we actually calculate the score for this for any population, and it is actually very similar to a logistic regression model. However, here this term makes it different from a standard logistic regression model. So here the beta 0k is the slope for each the intercept for each population, and beta 1k is the slope for each population, measuring their different effect on the health outcome, and h is just the logistic function. So as you can see, this model is slightly more complicated than a logistic regression model. And since we introduced those non-parametric terms, the estimation is also a bit more challenging. So in this project, we proposed two methods to estimate, which I will only briefly mention. One is a profile likelihood, another is the Bayesian model. So essentially, what makes it challenging to estimate is this slope term beta 1k. So if we don't have this one, it would reduce to a simple generalized additive model. However, we find like surprisingly, both methods give us very similar estimation performance. So now let's go to the enhanced application. Here we use the enhanced 2003 to 2006 data, and the outcome is a 9-year all-course mortality because we have their mortality data up to December 31st, 2015, which means that all of the participants in these 2 weeks had at least 9 years of measurement. So for the score component, we use MVP and ASTP. So you can argue that we can include more components, but then we find those correlations make the result not that interpretable. But this is for, this can be extended in the future applications. So for the non-score component, we adjust our model for age, smoker, weight, and education status, obesity, and self-reported health status. So actually, our model has one identifiability issue. So if you look at this term, this alpha j are unknown coefficient, and this beta 1k is also an unknown coefficient. So if we multiply this beta 1k by 1 and divide the alpha j by 2 and divide the alpha j by 2, then the term remains the same. So we have to impose a constraint that's the slope for the first population is -1. So after we impose this constraint, I just want to remind you that this would suggest a higher score value means a lower risk of mortality. So now let's take a look at the result. So on the top row, I'm showing the non-score component result. For example, we find for both men and women, there's a significant positive effect of age on the risk of all-cause mortality, which is very consistent with the literature. Oh, and also here we use the participants at age 50 to 85 to build up this scoring system. Also, being a smoker indicates a significant higher risk of all-cause mortality. We don't find a substantial difference across race and education levels, but there's significant effect on the self-reported overall health. So on the bottom left, I'm showing the score component. So actually here you can see for each component is a nonlinear function. And the left one shows that as you increase your MVPA per day, your score increase. And we find the most substantial increase up to 15 minutes per day. This number may be slightly lower than what you can find on the internet, but here we are focusing on a slightly older population. For the ASTP, we find that if you have the ASTP less than 0.3, your score should be high enough. And on the right, I'm just plotting the derivative to show the significance of those nonlinear functions. So after we build up the score, we also tested the predictive performance in addition to age, because we know that age is a very strong predictor. So as you can see, our model, so in addition to the age, if you include the score, the predictive performance actually increased by 0.06, which is quite substantial, and is better than any single component. So in summary, we built up a semi-parametric risk score for physical activity, and we use a novel generalized partially additive model, have two estimation approach. Our results suggest that in order to have a high good physical activity score for this older population, is recommended to have MAPA for at least 15 minutes every day and keep each active bout lasting longer than 5 minutes. This is related to the estimation of the ASTP. And here we also have several extensions. That's all. Thank you. Any questions?
It looks like Amal has a question. Is that Amal, want to ask it?
Yes, I'm looking at the chat.
Yeah, hey, that was excellent. I was just wondering about the cut points you're using for MVPA and whether they're outdated now.
Yeah, that
for older adults, could your results actually have even a stronger impact if you had a more appropriate cut point for older adults? And perhaps it may be the story of maintaining MVPA for at least 10 minutes or 8 minutes or something like that.
Yeah, that's a very good question. And to be honest, we didn't test it in this project, but I think it's related to the question like whether we would like to set a threshold like a scoring system, which do not depend on the population and do not depend on different outcome, or we want to set up a population-specific threshold. So here we use 100 and 2020 to the threshold is established by Troiano's paper, but I think it's worth exploring, like try different threshold. For example, in the UK Biobank, we actually find that we need to use those quantile matching to build up the threshold for this MVPA. And I think this is still an active research area.
Amal, one thing about changing the threshold is that you have to define what does it mean to be more appropriate, right? So the threshold is defined by Troiano is a threshold on essentially the number of Gs or the number of activity counts. And it has its problems, as you said, right? Because there are very few older people who would go to that level. But it also has its advantages in the sense that it's one definition for the entire population. So the reason why a lot of researchers said it's inappropriate is because you don't get enough variability in older populations, and therefore you lose some of the predictive power. But if we have thresholds for every single level, I think we have to think about exactly what everything means then, and we may open up an entire different thing about do we have a threshold for every age? Do we have a threshold for every BMI? And I think that's an area that will become difficult.
Well, again, let's thank so much both Rahul and Urja for two fascinating presentations, I think with real implications for older adults, and of course, everyone who has participated. As usual with these sessions, I want to give people who need to leave, your entree to do so now. We're coming up on 3 o'clock. But perhaps we can take, a few minutes just to see, whether there is any further open discussion, a thought that struck someone and you really don't want to lose it, or a thread that you saw connecting the three presentations. And so let's just give a moment and see whether there's any of those sorts.
Yeah, I want to say something. So I think we have showcased like a very nice work happening across 3 groups, but it would be great to spend a little bit of time thinking about like what's next and where the field is moving, right? Because we all have spent some time thinking about this in the past, but now the question is, what's next, right? Like what should we focus our effort on, right? And what would be the next very interesting things to look at? So anyone? I don't have any thoughts, I have only questions.
I love that question. And first of all, thank you, everybody. Just wonderful presentations. The level of work is just at such a, it's stellar. I'm curious about, the data that I look at is usually in the low intensity range, so we tend to think of activity generally as high to vigorous, moderate, and low intensity. And most of the work I do in populations I work with is really largely sedentary and low intensity. And so, I just wanted to bring that about because with the kind of brain power here, it would be interesting to think about what we can derive even in that signal in the low intensity range, if that makes sense.
It makes total sense. I mean, I also work more recently, I've been working on populations like who had stroke or COPD and we are used to, okay, 5,000 steps a day. And then you look at this population, it's 500 steps a day, right? If that. So very, very different populations. And what MVPA, means is a very different thing. I think that, we have to move a little bit away. I think that the problem with MVPA was that its definition was based on a threshold on that, nobody really knows what it means. But as long as you define MVPA, index a threshold that is well defined, right? For example, how many minutes are you above 20 steps a minute, right? So that's a very well defined thing, and you can go with that and adjust it to the population. But it's a very, very important question. How do we maintain interpretation across populations, while in the same time address the problems that we want, which is to have enough power to detect them, right? So I think that's something that will stay with us for a long time. I think that I see that interventions are very important. I think that longitudinal studies are very important. I think that the design of the experiment will be increasingly important. And I think that something we wrote a long time ago in a proposal that was never funded was this idea of the science as an interaction between smaller experiments and very large available data sets. I think it makes a lot of sense to take advantage of what is out there in terms of very large data sets to then better design and target our intervention. So I see a lot of papers and it's basically just this to go in the UK Biobank and check it. And some do turn out to be okay, others, no, they don't. So I think that we have to increase this interaction and in both directions, right? The smaller studies could inform what to look for in the larger studies, and the other way as well, where you go, let's take Alzheimer's disease or MS, because that's what I work on right now. You can easily do a matched case control study with thousands of people, and it takes an hour to do that. And then generate hypotheses for a really much more targeted study. So to me, that's an important, not just about my group or these groups that are getting together, but science in general. I think that's where we are going.
Yeah, I love that idea. And I think it's particularly important in older adults because the population one is, the sample one is able to assess can be highly selective, and so, in trials, there's a certain set of people who are willing to participate. And, so I think creative designs that use ancillary pieces will be very important to get the whole picture.
Chipre, maybe you should dust off that grant. Was that your U grant?
We were together on it.
I remember. I still love it. I love that.
Yeah, it was a fantastic grant that generated a lot of other grants. I'm not unhappy about the work we put into it, and today's presentations are many of them, just phenomenal. So if I sound bitter, I'm not. I mean, I'm very happy.
Well, it reminds me, to this point, Fine, you presented on the rebound and the restore, the spring study, and that kind of is a proof of concept of how these tools can be useful across a range of clinical and how do these summary measures translate. I think that was part of what you were saying, right? Is can we find measures, or maybe this is the way I interpreted it. How do we find measures that can translate across healthy and less healthy and clinical populations? Because our goal is to get the clinical population to look more and more like the healthy population as a metric of success of your intervention, right? And if you can show that you meet people where they are and say they're sedentary, but they're moving a little bit, and then you kind of think about how being more precise and being able to measure that movement or activity count, maybe then that can be used as a continuous outcome for an intervention design, trying to change behavior. That's kind of the way I would love to see that work go forward. So
Eleanor, I want to jump on what Michelle was saying, if I can take this opportunity. So first of all, thank you everyone for excellent presentations. I think we're getting closer to, I feel like there's this discussion about capacity versus behavior, and that putting capacity into context, right? So understanding what people can do versus what they do. And many people are maxed out. I mean, work that Jennifer did with BLSA data was, identified the distinction between doing what an individual's capacity was. And when you looked at the relationship with age, you found that there were many older, very old individuals who were actually working at max capacity, even though their total activity was low. And until we do that, prescribing activity rate needs to be more precise for the individual, especially in these sort of rehabilitation settings. And then I think we're in a better position to understand how well it can work and maybe when it doesn't work so well. And also to be able to understand activity, voluntary absence of activity versus limitations due to limited activity due to actual basic limitations. And then it becomes a much better tool for us. And I'll stop there.
Well, I guess if there's a pause, maybe this is a good opportunity to formally close the session, but I want to take one more opportunity to thank all of the organizers and participants. I agree that this was a fabulously stimulating session intellectually, and would really look forward to organizing more of these, rapidly moving opportunities for idea exchange. And so congratulations and thanks to everyone.
I agree. This is a highlight for me. Thank you, Brian, too.
Absolutely. And everyone, please have a great rest of your day.